Hi, I'm Joe Graydon.
I'm Terry Graydon. Welcome to this podcast of the People's Pharmacy, where we bring you the stories behind the health headlines.
This podcast is brought to you by Redix Industries, makers of Utterly Smooth Body Cream. 800-345-7339, on the web at uttercream.com. We count on antibiotics to cure us when we come down with an infection. Now superbugs are becoming resistant to these drugs. This is the People's Pharmacy with Terry and Joe Graydon.
What will doctors do when superbugs develop resistance to the antibiotics of last resort? That day has already arrived. What can be done to treat infections in a post-antibiotic era?
Is there anything we can do to slow the rise of superbugs? Has our overuse of antibiotics to treat every ear infection contributed to this problem? What about drugs used to fatten farm animals? Will the FDA do anything to discourage overuse?
Coming up on the People's Pharmacy, we find out about superbugs and how viruses might help fight them.
First, the news.
In the People's Pharmacy health headlines, morning sickness is a common occurrence with early pregnancy. Although it can make women feel miserable, new research shows this cloud has one silver lining. Women who experience nausea and vomiting associated with the onset of pregnancy are 50 to 75% less likely to lose the baby before the pregnancy is completed. The research included almost 800 young women very early in a confirmed pregnancy. All of these women were in the study because they'd had previous pregnancies that didn't end well. During this trial, almost 1/4 of the pregnancies ended in miscarriage. That's a much higher ratio than usual. But women who experienced nausea alone or with vomiting were less likely to experience this outcome. No one knows why morning sickness is so common with pregnancy, but the authors hypothesize that viable placental tissue to support the embryo's growth may release hormones that can cause nausea. They caution, however, that women who aren't having morning sickness should not worry that there's something wrong with the pregnancy.
Lowering dangerous LDL cholesterol is a primary focus for people who want to lower their risk of a heart attack. Sometimes people who have side effects on statins wonder if getting their cholesterol down through other means will work just as well. Until recently, doctors did not have an evidence-based answer to that question. Now, a meta-analysis compared statins and other cholesterol-lowering therapies. The aim was to see how much such treatments reduced cardiovascular events such as heart attacks and strokes. There were 49 trials in the meta-analysis with 312,000 volunteers. The studies included in the analysis used various methods to lower cholesterol. First, statins. Second, diet or drugs like Questran or Welchol. Third, medicines like fibrates or niacin. And fourth, the new PCSK9 inhibitors. What mattered most for preventing heart attacks and similar problems was how much people managed to lower their LDL cholesterol. The authors point out that statins are a relatively easy way for most people to move that needle a lot. PCSK9 inhibitors may end up having a big effect, but we won't know for sure until the studies on these new drugs are completed in 2017. In the meantime, the main message is to keep cholesterol under control to reduce cardiovascular risk.
Kidney stones form in the kidneys when calcium and other minerals clump together. They eventually move into the bladder and then into the urethra, where they can cause excruciating pain as they pass through. Anyone who's ever had a kidney stone would be extremely grateful to have a way to pass them more readily. One patient reported to his doctor that a trip to Disney World and a ride on the Big Thunder Mountain Railroad roller coaster resulted in his passing 3 such stones. The curious urologist decided to investigate. He built a model kidney, filled it with urine and kidney stones, and headed for Disney World. Sure enough, sitting in the last car of the roller coaster resulted in passing the stone 64% of the time.
How good is the research on artificial sweeteners? Consumption of artificially sweetened beverages and foods has increased dramatically over the last 40 years. We often hear that these compounds are extremely safe and helpful, but a careful analysis of 30 studies shows that conflicts of interest are common and may distort the reported findings and conclusions. Reviews carried out by scientists whose conflicts aligned with the artificial sweetener industry were far more likely to have favorable results and come to favorable conclusions than those not sponsored by industry. Those conducted by scientists whose conflicts aligned with competitor industries, such as water or sugar, reached unfavorable conclusions. The investigators conclude that such conflicts may undermine the quality and transparency of public health evaluations that are reliant on these reviews.
The CEO of the company that makes the EpiPen auto-injector was grilled on Capitol Hill a week ago. She justified the $600 price of a 2-pack on the grounds that Mylan only makes a $100 profit. The majority goes to middlemen. On further investigation, however, it seems the actual profit was 60% higher, at $166. And that's the health news from the People's Pharmacy this week. Welcome to the People's Pharmacy. I'm Terry Graydon.
And I'm Joe Graydon. Superbugs that have developed resistance to many common antibiotics pose an increasing threat to public health. 2 million Americans fall ill each year due to infections from hard-to-treat bacteria. Tens of thousands die when the prescribed antibiotics didn't work.
Recently, the UN General Assembly passed a declaration with the goal of slowing antibiotic resistance worldwide. Countries are being asked to come up with a plan over the next 2 years to protect the ability to use antibiotics to treat serious infections in humans. What should we know about antibiotic-resistant superbugs?
To find out, we turn to Dr. Barbara Murray. She's the J. Ralph Meadows Professor of Medicine and Director of the Division of Infectious Diseases at the University of Texas Health Sciences Center at Houston. She's also past president of the Infectious Diseases Society of America.
Welcome to the People's Pharmacy, Dr. Murray.
Thank you.
Dr. Murray, you recently had an opportunity to testify before the House Energy and Commerce Committee's Health Subcommittee, in which you suggested we may be facing a post-antibiotic era with mortality rates for infections increasing. You know, most Americans have no idea what you're talking about. What would a post-antibiotic era look like?
Well, it would mean that infections that are easily treated today might not be easily treated tomorrow. And in particular, we're seeing this in certain settings, the hospital settings, so we could see, we're already at the stage where some people with cancer that have, they're immunosuppressed, they have had their bone marrow transplant, they have no immune system to fight infection, they are getting infected with bacteria that we cannot treat. They're basically resistant to everything that's commercially available. Now, a lot of people will say, well, you know, my chances of getting cancer are low, which is not necessarily true, but we see it in other settings. In the hospital, and I'll tell you some we see outside the hospital, somebody in a bad car wreck comes into the hospital and has a months-long course of everything's broken and they have tubes coming out of every part of their body. And they're on multiple antibiotics, which then selects for more and more resistant antibiotics, and they too can become resistant to the bacteria, can become resistant to everything. In my testimony, I mentioned a patient that I did not directly take care of, but was taken care of by a colleague of mine who had a prosthetic knee replacement. That's something I'm facing. A lot of people are facing that in the future, and with the aging population, there are going to be more of them. And there's a low infection rate, but this unfortunate person had got infected with one of these gram-negative bacteria that's very resistant, and it then developed resistance to every antibiotic that was tried, and he eventually had to have an above-the-knee amputation.
Dr. Murray, why is there so much antibiotic resistance now?
Well, There are a lot of reasons. One is that, of course, the bacteria have been around longer than we have. They also multiply and mutate at a very fast rate. So whereas it may take us, thousands of years to evolve, they may be able to do it in a few weeks. So that by mutating, they may be able to mutate to resistance to certain antibiotics. And they're always mutating, and it's by chance and just random. But if one of those mutations causes resistance to an antibiotic that patient is on, then that bacterium survives. It's really easy in a test tube if you see it. You have, a million bacteria and only one is resistant. You treat it with antibiotics, and all of a sudden most of the bacteria die off, so the counts go down until that one has time to multiply, and by the next day, it's now a million of it. So that's the numerics of it. You take one, and it becomes a million in the presence of antibiotics. The other thing is that bacteria transfer a lot of their genes. They have these mobile genetic elements that can transfer from bacterium to bacterium, kind of like for us, we spread flu from each other, one to another, or any infection. They can spread these plasmids or other mobile elements from one bacterium to another. So if an enterococcus develops resistance to vancomycin, that enterococcus has the potential to transfer that, and it's a very complicated 8-gene sort of operon it takes. It's not a simple thing, but it's got the vancomycin-resistant enterococcus has the whole thing and can transfer that all in one piece to Staph aureus, and that's exactly what has occurred. There are rare strains of vancomycin-resistant Staph aureus, and they got the genes from enterococci. So that's sort of the genetics of it, and then our use of antibiotics, as I mentioned, just allows the resistant bacteria to have this selective advantage. It's the survival of the fittest. They're in the presence of antibiotics. They're the ones that become dominant. And then the fact that we spread antibiotic-resistant bacteria from one to another. So in the community, in a daycare center, one child has a resistant strep in their throat or pneumococcus in their nose, and they are touching each other and spreading it all around. We did a study years ago with daycare centers and trimethoprim sulfa or Bactrim resistant fecal E. coli, and it was a diapered age population, and we just wanted to see what happened in a daycare center. And in one of the daycare centers, the same exact strain by DNA fingerprinting was found in like 70% of the children because they're diapered age and their diapers leak and they spread things. So there's spread. In the hospital setting, we also spread bacteria, and I'm sorry to say that happens, but if you look at the example with Ebola, contact precautions where you just wear the gowns and gloves is supposed to prevent spread of things like Ebola, just as it's supposed to prevent spread of things like antibiotic-resistant bacteria. The problem is the degree and stringency of adherence that has to be carried out cannot be done in a practical setting. So what we saw with Ebola is now it takes, multiple pairs of gloves. You have to have an observer watching somebody come out. It's often the coming out of the coverage that the doctors and nurses are in that leads to contamination of their own skin or the environment. So despite best efforts to alcohol hands, wear gloves, wear gowns, etc., we spread the antibiotic-resistant bacteria in the hospital just as Ebola spread in those few cases.
Dr. Murray, Americans have this wonderful optimism about technology, that there's a solution for just about every problem right around the corner. And we recently heard about a discovery of a new compound, Teixobactin, which has been suggested, oh, wow, this is something new, this is something wonderful, it won't have antibiotic resistance, it'll be at least as good, if not better, than vancomycin, it'll treat difficult methicillin-resistant staphylococcus. But is it really that good? And, what does the future hold?
It's hard to say if that particular antibiotic is going to be that good. It has to pass long-time toxicity issues. It has to, go into man. We've said before that resistance is not going to develop, and it did to vancomycin, and people say, well, it took 30 years. Well, it wasn't used very much the first 30 years. It really developed fairly quickly after the era of when we started using it a lot. it looked that a drug, I have two comments about this. The drug looks promising. However, staph is not really the biggest problem right now. We just had 2 or 3 antibiotics approved for staph already in the past year. And there are already some that are very good for staph out there. It's a terrible bug, and it can kill people within 24 hours, but it's not always because of the resistance. I mean, if it's mistreated, yes. But, I mean, we do have pretty good drugs for staph already. My excitement about this is actually the technology and the fact that it uses, what it did was capitalize on what was the original and best way that we've ever had for finding antibiotics. They find them in nature. The key in this paper is that they've developed a technology to find the antibiotics in those fungi or organisms that are not cultivated, don't grow on routine laboratory medium. And it's about 99% of what's in the soil, which is where most of our antibiotics have come from, or organisms and bacteria. About 99% don't grow in routine culture medium. So we're missing the screening for 99%. So basically what they are able to do with this technology is increase 100-fold, in essence, our ability to now find new antibiotics. And I think that's the exciting part. One more staph antibiotic I'm not that excited about. What we need are antibiotics for the gram-negative bacteria like Pseudomonas, Acinetobacter, these carbapenem-producing NDM penicillinase, beta-lactamase bacteria that are our big scourge at the current time. But presumably this technology can be used to discover if there are new antibiotics against those organisms as well.
Dr. Murray, is there anything our listeners can do as individuals to help reduce the problems of antibiotic resistance?
Well, I think there are a couple. One is if the doctor says you don't need an antibiotic for this cold, don't press them on it. Many people feel like if they go to a doctor and don't get an antibiotic, they haven't gotten their money's worth. And antibiotics do not work for the common cold. They do not work for viruses. So there's been a big campaign, and a lot of pediatricians' offices now have signs trying to explain why we're not giving your child antibiotics for what appears to be a virus. Because the more antibiotic use there is, the more antibiotic resistance is going to emerge.
Dr. Barbara Murray, thank you so much for talking with us on the People's Pharmacy today.
You're welcome. Thank you.
You've been listening to Dr. Barbara Murray. She is the J. Ralph Meadows Professor of Medicine and Director of the Division of Infectious Diseases at the University of Texas Health Sciences Center at Houston. She's also past president of the Infectious Diseases Society of America.
A couple of weeks ago, the Natural Resources Defense Council and 6 other advocacy groups delivered a petition to the FDA. They want the agency to stop allowing the widespread routine use of antibiotics to promote growth or prevent disease in poultry and livestock. The hope is that cutting back on unnecessary antibiotic utilization for food animals could reduce bacterial exposure and the evolution of resistance.
After the break, we talk with Dr. Alan Green and get his perspective on antibiotic therapy.
What would it mean to live in a post-antibiotic era?
Why do doctors sometimes prescribe antibiotics unnecessarily?
What are the downsides to antibiotics besides increasing resistance? How is the microbiome affected?
Stay tuned also for our chat with a scientist who has learned how viruses can be harnessed to overcome superbugs.
You're listening to The People's Pharmacy with Joe and Terry Graydon.
If you like the information you hear when you listen to The People's Pharmacy, you may also enjoy our book published by National Geographic. It's The People's Pharmacy Quick and Handy Home Remedies, available online at peoplespharmacy.com or at your favorite bookstore. Welcome back to The People's Pharmacy. I'm Joe Graydon.
And I'm Terry Graydon. The People's Pharmacy is brought to you in part by Red X Industries, makers of utterly smooth creams and lotions that deeply moisturize to make dry skin feel soft and smooth. 800-345-7339. Online at uttercream.com.
If you would like to order a CD of the show, you can call 800-732-3738. 800-732-2334. Today's show is 1052. That number again, 800-732-2334, online at peoplespharmacy.com.
We're talking today about bacteria that have developed resistance to life-saving antibiotics. Will there come a day when we can no longer treat infections like gonorrhea, pneumonia, or meningitis because the medicines we've relied on for decades no longer work?
Our guest in this part of the show is Dr. Alan Green. He's the founder of drgreen.com and a pediatrician in private practice. His books include Feeding Baby Green, Raising Baby Green, and From First Kicks to First Steps. In 2010, he founded the Whiteout movement to encourage families to offer children regular food instead of highly refined white rice cereal when they begin to eat.
Welcome to the People's Pharmacy, Dr. Green.
Thank you. Great to be with you, Terry and Joe.
We love having you on the People's Pharmacy, Dr. Green, to bring a little perspective on, in this case, antibiotic therapy. You know, we've looked for years at antibiotics as magic bullets, and they were prescribed routinely for children who had ear infections. I remember our kids, anytime their ears start to hurt, out would come the bubblegum-flavored amoxicillin.
Indeed.
And now, wow, we've changed the way we think about this. Could you help us understand what's going on?
Sure. Well, first of all, antibiotics were something of a magic bullet. When penicillin was first introduced to treat people with bacterial infections back in 1942, it was a new era. Things that could easily get very bad and kill people were easy to treat. But it turns out that bacteria very quickly are able to respond. When antibiotics kill some of the bacteria, the ones that are left are a little bit more resistant, and over many generations, very quickly it changes. And as soon as 1945, 20% of the staph bacteria found in hospitals were already resistant to penicillin. So there was this steady stream of new antibiotics, new classes of antibiotics, but for most of our childhoods, most of the antibiotics worked pretty well still. But we have crossed a line in 2008 where this process of resistance accelerated so much that we have now found bacteria that are not only able to resist antibiotics, but that can live on antibiotics as their only food. So it's time to rethink how we treat many of the common childhood infections. Now, I should mention, by the way, that this stuff that they found, the antibiotics that could live, the bacteria that could live on antibiotics as their food source, it could eat antibiotics for lunch, was not found in a hospital. Bacterial resistant used to be a hospital problem. It wasn't even found in a home. It was found in the soil. Now, that's another big shift that's taken place in our lifetime. Not that long ago, back in 1998, most of the antibiotics used in the United States were used to treat people who were sick. But now 80% of all antibiotics in the U.S. are used as growth promoters in animals, really changing the picture. And the most resistant bacteria were found in soil.
Well, you know, Dr. Green, I find it just amazing, this concept that antibiotics become food for bacteria. I mean, that's just bizarre. But we heard something just recently that one form of bacteria can transfer its resistance to antibiotics to different kinds of bacteria, and they can kind of learn this resistance process through their genes. I mean, that's really scary.
It is. It's sort of hot swappable. Imagine you had a thumb drive and wanted to move a program from one computer to another. Well, bacteria kind of have that. They have little segments of their genes. They can take something they've got encoded, the ability to resist an antibiotic, and swap it with all their neighbors very easily.
Now, Dr. Green, we've heard that all of this is pointing to a post-antibiotic era. What would that mean?
Well, what that would mean, if we enter a post-antibiotic era, which the World Health Organization has now said, unless we take significant coordinated action, we're headed that direction. That would mean that simple infections and simple wounds that used to be easy to treat could suddenly be major life-threatening conditions again. Now, I'm actually somewhat optimistic, because even if we do have larger epidemics of resistant bacteria, and they already are happening, by the way. There's about 2 million infections in the U.S. each year with resistant bacteria, about 23,000 deaths each year in the U.S., 25,000 in Europe. But if we have really significant ones, I do think there will be a concerted effort. We're not starting from scratch. People will figure out new classes, new ways of combating bacteria. But we could be in for some tough years in between.
Dr. Green, we've seen studies, for example, one recently in pediatrics, that show that doctors are still prescribing antibiotics for a lot of infections that are probably viral. What can be done about that, and why does it continue?
So a lot of times when people think about infections, they think bacteria, virus are kind of similar to each other, but they're entirely different species of different classes of organisms. If bacteria happen to be the size of an elephant, a virus would be the size of a mouse. So they're really different. And antibiotics that work for bacteria do not help at all for viruses. But when people are sick, and sometimes, quote, just to be safe, someone will prescribe an antibiotic, and just on the off chance there may be some bacteria there. And this doesn't make the situation better. It does make it worse. And it happens just, as I think, because people are rushed. They want to be able to give people something to help them help. And often, as well, people want a prescription when they go see a doctor and they have especially a sick child with a fever. They want something to be done.
So you have to deal with this on a routine basis, and when you run into a family that says, well, come on, Dr. Green, can't you just prescribe something? How do you respond?
Well, I explained that my goal is to get this child well, and that one of the best things for their long-term health is to cultivate their immune system. We'll watch over them carefully and protect them, but not to disrupt that by unnecessary antibiotics. One of the things online, when I answer questions at drgreen.com, I have a sheet that I tell parents, print this out and bring it with you to the doctor. And it starts with the question, is there a safe way to treat this without antibiotics? And I think it's a question every parent should learn.
You know, we talked about the magic bullet idea, and I think most people assume antibiotics have this huge benefit and virtually no downside, no risks. And yet, when children, or adults for that matter, are put on antibiotics for, long periods of time, I think about adolescents who are put on tetracycline, for example, not for weeks or months, but years, that it's conceivable that antibiotics actually can disrupt the bacterial flora and fauna of the digestive tract. And I wonder what those consequences might be. Are we beginning to find that there is a downside?
We are learning that there's downsides to it. So it's not just about antibiotic resistance and losing them for when we need them, but our bodies have this whole rich ecosystem of beneficial bacteria that help us digest our food. They're an important part of our immune system. They're an important part of who we are. I mean, 90% of the cells in and on our body are bacterial cells, beneficial bacteria. And when they're disrupted by antibiotics, they can cause disease. They can cause nutrient deficiencies. They can trigger allergies. And perhaps there's some early evidence that it can contribute to obesity, getting antibiotics. We know that the antibiotics are used because in agriculture because they fatten up the cattle.
Now, if we turn back the clock 20, 30 years when our kids were young, and we took them to the pediatrician, and there was a little redness or swelling in the ear, or perhaps even pain. As I mentioned earlier, out comes the prescription pad for amoxicillin primarily. But what about today? What are pediatricians being told when it comes to ear infections, and what are you doing in your practice?
That's a great question, because not only the situations where antibiotics don't work, like viral infections, that is beginning to shift and they're being used less. But we've now learned that even situations where antibiotics do work, like on bacterial ear infections, it's often better to wait and watch carefully and see where things go over 48 hours, because many times kids will get better on their own. And if they do get better on their own, they're better off. Their immune system is primed. They're less likely to get another ear infection soon. And if they do, it's less likely to be with resistant bacteria. So now there are guidelines for which ear infections it's okay to watch and wait and take care of them and solve the pain in the short run, and which ones are concerning enough that they do need treatment right away. But it's no longer just because antibiotics would work, are they a good idea?
Well, how would a parent know, for example, if a child really did need a prescription for antibiotics because the ear infection was getting worse?
Well, I do think that an ear infection, and you can generally tell if it's an ear infection that might need antibiotics because the child's in pain. And if there is an ear infection, I think that's a great time to be in touch with a doctor, because you do want someone to watch and wait through this with you, to be looking for signs of concern. But in general, if the child is 2 years old or older, it's less likely to need antibiotics, even if it's a significant ear infection. And if they're under 2, it's less likely to need antibiotics if it's not accompanied by really severe pain and not accompanied by a significant fever, like 103.
Who's responsible for antibiotic resistance?
For a while, we were thinking that physicians are responsible for overusing antibiotics, and certainly we should share some of the blame for that. And then I've heard people say that parents and patients are responsible because they're asking or expecting antibiotics. And I think it is wise for patients to be educated to ask, is there a safe way to do this without the use of antibiotics? Recently, my focus has been on agriculture, since 80% of the antibiotics are used in agriculture, and I do think there's significant responsibility there. And when you reverse it, resistance improves. But there is another source that I've just become aware of recently that may be an even bigger piece of the puzzle, and that is antibiotic resistance spreading in our waterways. There's just a new study in one of the Nature journals looking at the waterways, looking at what's flushed down our toilets and what runs off from farms, gets in the waterways and spreads this wave of resistance along the course of the water. And it turns out that pharmaceutical factories where they're making the antibiotics, a third of all antibiotics are made in India, more than that are made in China. They've been now looking at the water near the manufacturing plants. And in one, for instance, at Hyderabad, they found enough Cipro, a strong antibiotic, in the water, that it was at higher levels in the water near the plant than in a human's blood when they're being treated for a serious infection.
Wow.
There was enough, they found, to treat all of the population of Switzerland for 5 days with Cipro in the water there.
That's pretty scary.
It is. So dealing with antibiotics in the water system may be another really important piece of the puzzle in getting rid of resistance.
You've been listening to Dr. Alan Green, founder of drgreen.com and author of Feeding Baby Green and Raising Baby Green. Dr. Green is a pediatrician in private practice in the Bay Area of California and a founder of the Society for Participatory Medicine. We apologize for the sound quality. Dr. Green spoke to us from his home office in Emeryville, California.
What do we mean exactly when we talk about bacterial resistance? For a very different perspective, we turn now to Professor Paul Turner. He's chair of the Ecology and Evolutionary Biology Department at Yale University. He's also a member of the microbiology faculty at the Yale School of Medicine.
Welcome to the People's Pharmacy, Dr. Paul Turner.
Thank you. It's a pleasure to be here.
We are delighted to have you with us today. Dr. Turner, we keep hearing a lot about bacterial resistance, and it's a growing problem. From your perspective, how bad is it?
Well, I guess I would have to say it's pretty bad. We really have benefited a lot within the last roughly 75 to 80 years in using chemical antibiotics to treat bacterial infections. And as most of your listeners know, the efficacy of these drugs is just increasingly waning in efficacy. And the reason is that bacteria have evolved resistance to most of the frontline drugs and even many of the alternatives.
So we really are talking about evolution here, huh?
Yes, we are. A lot of people have characterized this as just after Fleming discovered penicillin, and then you had widespread production of chemical antibiotics not that long after that to administer in people. When you couple that with the fact that very many antibiotics are used in agriculture, we're essentially doing a large uncontrolled selection experiment. And the result has been widespread and expected response on the part of bacteria to evolve resistance.
Now, what do we mean when we say resistance?
So what we mean is that genetically the bacteria have changed so that they are truly able to withstand these drugs that we throw at them. And the way I like to describe it is it's particularly alarming because it seems to be occurring largely without any cost. In other words, what we care about in evolutionary biology is the fitness cost. And it's almost as if you can consider bacteria are collecting these like trading cards, and they're not paying any cost for carrying around their large collection of trading cards with them. They're able to withstand very many antibiotics, and increasingly so.
What sort of cost would you expect to see?
Well, often in evolutionary biology, if you do see the acquisition of a new trait through evolution, it's kind of the sense of you can acquire a new adaptation, but often it limits your ability to do something else that you might be doing, or to suffer a reduced ability to do some other trait. And what I'm saying is that bacteria evolve resistance, and it doesn't affect their growth.
Now, we hear about superbugs, and there have been a number of, almost call them exposés on the evening news, where you hear about bacteria that are really, really dangerous. I mean, these are killers, and we've kind of run out of bullets. They are, if they get loose in hospitals or other health institutions, they become extraordinarily difficult to deal with. Can you give us a little more insight on these superbugs?
Yes. So I guess if somebody asked me how would I define a superbug in terms of evolution, it's a very scary beast for biomedicine because there are many bacteria that are very dangerous for humans. Fortunately, not that many of them relative to the total diversity of bacteria in the world. But if you're talking about bacteria that can effectively get in the human body, cause enough damage to kill, the problem with superbugs is that they're able to do this, and they seem to be increasingly able to withstand the drugs that we throw at them. So to me, they are kind of superorganisms in the sense that they can thrive doing what they can as pathogens within the human body, and they're super at it in the sense that it's very difficult and increasingly failing in our ability to control them.
You're listening to Dr. Paul Turner, Chair of the Department of Ecology and Evolutionary Biology at Yale University.
After the break, we'll find out if we are likely to run out of antibiotics since so few are now being developed.
Find out about a possible alternative to antibiotics, bacteriophage therapy.
What's the history of phage therapy and its development?
Could a virus discovered in a Connecticut pond offer a new weapon against antibiotic resistance?
You'll also learn about the long-term implications of Dr. Turner's research using the power of evolution against bacteria.
You're listening to The People's Pharmacy with Joe and Terry Graydon. If you value the health information you get when you listen to The People's Pharmacy, consider subscribing to our email newsletter. You'll get the latest health news and information on upcoming podcasts delivered to your inbox twice a week. Look for the link at peoplespharmacy.com. Welcome back to The People's Pharmacy. I'm Terry Graydon.
And I'm Joe Graydon. The People's Pharmacy is brought to you in part by Red X Industries, makers of Utterly Smooth Foot Cream with shea butter, helping dry, cracked feet feel utterly soft and smooth. The number's 800-345-7339, online at uttercream.com.
If you'd like to order a CD of today's show or any other People's Pharmacy broadcast, you can call 800-732-2334. Today's show is number 1052. The number 800-732-2334 or online at peoplespharmacy.com.
Antibiotics have long been perceived as our defense against life-threatening infections, but many nasty bacteria are developing resistance against important antibacterial drugs. What will we do in a post-antibiotic age? Our guest has some novel insights on how a discarded therapeutic approach might offer hope for the future.
We're talking with Dr. Paul Turner. He's chair of the Ecology and Evolutionary Biology Department at Yale University. He's also a member of the Microbiology faculty at the Yale School of Medicine.
Now, I think Americans have this perspective that we can always come up with some new trick, some new technology. There's something just around the corner that when all else fails, we'll pull something out of the hat that will save the day. And yet there are some microbiologists who are saying, we just haven't invested in antibiotics the way perhaps we should have. They're not the billion-dollar drugs that the pharmaceutical industry is so excited about, the kinds of drugs that people take on a daily basis, perhaps for the rest of their lives, statin-type medications, proton pump inhibitors for indigestion. And we're going to pay a price in the not-too-distant future because we just may run out of new developments in antibiotic therapy. Is that fear justified?
I think that fear is pretty justified, in my opinion. So the problem I see is that pharmaceutical companies, they did produce antibiotics, and they would look for synthetic antibiotics, those that are unknown in nature, but they are created and made to look a lot like existing antibiotics, and they will function in killing bacteria. So the problem is, anytime an antibiotic is used as a large-scale drug, there is this selection for bacteria to become resistant to them. So in essence, what I'm saying is there's not very much money left in it for pharmaceutical companies, at least as far as I could tell. So they could bring these into production. They could get, say, approval from the U.S. Food and Drug Administration to safely put them into people. And by the time they've spent all that money, which is a considerable amount of money that they've sunk into this, if those drugs then quickly fail, and in the eyes of a business, a quick failure would be, say, within a decade, then they've got problems. They've invested in something that doesn't work. And pharmaceutical companies are out to make money, and understandably so. And the question is, why would they sink any further money into large-scale production and new discovery of synthetic antibiotics if they're only going to fail ultimately? So I think really they've become less interested.
Dr. Turner, as we understand it, you and your colleagues and your graduate students have been working on an alternative to antibiotics, something called bacteriophage therapy. Can you tell us about it, please? What is it?
Sure. So phage therapy, for short, is something that was largely developed in the former Soviet Union and in Eastern Bloc nations such as Poland, around about the time that we were in the West investing in chemical antibiotics. And in fact, phage therapy was tried out long before Fleming even discovered penicillin. What phage therapy is essentially a phage is a virus that is specific to a bacterial species. And if you find the right phage, then you could safely put this in the human body or apply it as a topical, say on a wound on the outside of the human body, and it will attack and kill the bacteria that it is specific to infecting. And it doesn't seem to cause any harm to a human and alongside that. So the point is, it could be used as an alternative strategy for killing pathogenic bacteria, has been used in the past successfully, and now that we've run out of options, we are looking again at this old alternative, phage therapy, but it really needs to be updated.
Now, if I understand what you just said, these bacteriophages, short name phage, Phage, they're viruses, and these viruses actually kill the bad bacteria. Is that right?
That's correct.
Wow. It's like an ecology. There are viruses that attack bacteria and kind of keep them in check. And do they exist in our bodies? I mean, do we actually have bacteriophages floating around kind of controlling the environment?
Absolutely, you have bacteriophages in the air you breathe, the water you drink, and the food you eat. And they have evolved on this planet long before us, around the same time probably as bacteria. My point in saying that is that humans ingest phages all the time, and in studies that look to see whether there's any immune response in humans, there tends to be none. That doesn't surprise me, because essentially they are part of our environment, and if we were constantly reacting to them with our own immune systems, we'd be in trouble. There would be a lot of wasted effort. So they can, in theory, control bacterial population sizes in, say, the ocean. So we know that cyanobacteria are responsible for a lot of the world's photosynthesis, and you find so-called cyanophages alongside them in the oceans. And many people believe that they are kind of keeping the numbers of cyanobacteria more or less in check through all of this kind of battling it out within the oceans for phages infecting bacteria and bacteria trying to escape them.
Dr. Turner, you mentioned a little earlier that in the former Soviet Union there was interest, and I think also in Poland, that they actually had something called bacteriophage therapy. How did they do that? What was going on?
So they had the idea that this was after phages had been discovered and described. They had the idea that if this specificity does exist between phages and certain bacterial species, you should, in theory, be able to go and find in nature a phage that would do the job of treating a bacterial infection in humans. And they were correct. So you go to places where phages are extremely numerous, and that would be in, say, fresh water or in soil systems. And if you have an efficient way of sifting through that material and finding a phage that will target and kill the bacterium of interest, then you've got yourself a naturally occurring drug that you could put into people. And maybe just to finish that thought, the listeners should know that this was before a lot of easy genetic characterization was possible. So it was essentially drugs that were found in the environment and administered into people largely effectively and safely. But in essence, it was really largely the use of uncharacterized drugs, which does make biomedical researchers, especially in the West, pretty uncomfortable.
Dr. Turner, we understand that you found something floating in a Connecticut pond. Now, of course, you're in New Haven, Connecticut, so obviously it would be in Connecticut. But this thing, whatever it was you found in the pond, might be a new weapon against antibiotic resistance. Please tell us more.
I'd be happy to describe it. So taking a step back to the old idea of phage therapy, we wanted to update it. And the way we wanted to update it is to find phages that would target and kill some bacterium that is a pathogen. But we know as evolutionary biologists that it is inevitable that anything you use to kill bacteria, if it is within their means to be selected to resist it, it will happen. They're very capable of doing this, as we've seen with traditional antibiotics. So we searched for phages that would attach to bacteria and kill them, and in the process, this would cause the bacteria to respond through evolution and to become more sensitive to antibiotics as a result. Essentially, we were trying to cause a genetic trade-off. That's what we call this in evolutionary biology, where you do force an organism to deal with an environmental challenge with the hope that it gives up something valuable in response. And in this case, the thing it would give up is resistance to antibiotics, and instead they would become sensitive. We tried engineering this in the laboratory. There are ways to do that, but we decided, let's see if there are any uncharacterized phages that simply attach to bacteria in this useful way for humans. And indeed, we found one floating in a lake called Dodge Pond in Connecticut.
Tell us more about it. What does it do, and how does it do it? And what are the implications?
So the way that this phage attacks bacteria is it attaches to a protein on the exterior of the cell that's part of a complex of proteins that pump antibiotics out of the cell. These are called efflux pumps. They've been described for a long time in bacteriology. So part of the problem of, say, trying to treat Pseudomonas aeruginosa as an opportunistic pathogen is that it's hard to get the antibiotics into those cells anyway, and when they get in, they get pumped right back out by this complex of proteins. So what we looked for were phages that naturally attached to the outside protein complex, and we assumed that if the bacteria had to deal with this phage in their environment to become better at withstanding their attacker, they might just change that protein complex so it becomes less effective at pumping out antibiotics. And that is exactly what happened. Fortunately, it seems to be a general thing. We took strains of that bacterium, Pseudomonas aeruginosa, that are just found in natural places like estuaries. We took some that had been isolated from household sink drains in Louisville, Kentucky, and we also took others that were taken directly from clinical samples that had been in patients. In all of these cases, the phage exerted the right type of selection on these bacteria, that the bacteria had to respond to their attackers by changing their efflux pumps. As a result, they are now more sensitive to antibiotics that are essentially useless in controlling them. So you have to think of it as a combination of administering this phage and the antibiotic should be a way that you can kill not only the bacteria if the phage do the job, but you'll also kill the bacteria that evolve resistance to the phage because they've become sensitive to the antibiotic.
Well, that sounds really exciting. Is it close to being able to be used, for example, in a clinical situation?
What we seek to do is a large clinical trial. For example, cystic fibrosis patients unfortunately suffer often from Pseudomonas aeruginosa infections. Others as well, other people who are immune compromised, those who have severe burns. And we would like to have a large number of individuals to treat with either the phage or not, and to, at a broader scale, look at the efficacy. Already, we are placing this phage with FDA approval inside human volunteers who have Pseudomonas aeruginosa infections that are multi-drug resistant. They essentially are out of options for how to control those infections. They are willing to undergo experimental therapy. Our hopefully soon to be published paper on the first case worked, and we were able to kill a bacterial infection around a heart in a patient who had suffered this infection in association with an aortic arch replacement surgery.
Dr. Turner, what are the long-term implications of your research, not just for Pseudomonas, which is a nasty bug, but for lots of other bacteria that also are on the verge of perhaps developing resistance to our best and latest antibiotics? I mean, is there a hope that bringing phage therapy back into mainstream medicine might reverse this trend?
That is entirely our hope. That's an ambitious hope, but it is what we seek to do. So we are currently seeking and looking for phages that work in the same way as the one that we characterized for Pseudomonas aeruginosa, using a lot of other target bacteria. In my opinion, it's a big phage world out there, and essentially that teeming biodiversity is something that we should be able to tap into in a kind of a natural product pipeline, that we could find these phages, we could characterize them, we could test them for efficacy and safety, wanting to make sure that nobody would be harmed in the process. But it would be wonderful if we had this new alternative to use against bacterial infections, and not just one opportunistic bacterial species, but there are very many of them that cause morbidity and mortality in humans. So in theory, we could find very many phages that are naturally occurring and use them to really revive the antibiotics that are no longer useful, because in combination with phages like these, if they make the bacteria sensitive to antibiotics, then we really don't have to establish a new pipeline of antibiotic discovery. Instead, we use this pipeline of phage discovery to use what's already in our drug arsenal.
Well, as you've described it, there's the expectation that the bacteria will ultimately develop resistance to the phage, but that the way that the phage operates will actually then make those bacteria able to be treated in some other way.
That is correct. So we are trying to find the weak link for these bacteria in their expected evolutionary response. So the thing to remember is that for any opportunistic bacterium that can be a pathogen, there are myriad other ways that it can go about a happy lifestyle on this planet outside of the human body. And they are just taking advantage of entering one environment in which they could grow. In a sense, we're trying to force them out of these environments that they might opportunistically enter. And when they do, we've got something that can effectively control them, the use of these phages and the reversion to antibiotic sensitivity in the bacteria.
That is very exciting. Dr. Paul Turner, thank you so much for talking with us on the People's Pharmacy today.
Oh, thank you for having me.
You've been listening to Dr. Paul Turner. He's chair of the Ecology and Evolutionary Biology Department at Yale University. He's also a member of the microbiology faculty at the Yale School of Medicine. Dr. Turner is an associate editor for the journal Evolution, Medicine, and Public Health.
Lynn Siegel produced today's show. Al Wadarsky is the engineer. Dave Graydon edits our interviews.
The People's Pharmacy is produced at the studios of North Carolina Public Radio, WUNC.
The People's Pharmacy theme music is by B.J. Liederman.
The People's Pharmacy is made possible by Redix Industries, makers of Utterly Smooth Extra Care 20 cream with 20% urea for extra skin protection. The number's 800-345-7339. The website is uttercream.com.
If you would like to order a CD of today's show or any other People's Pharmacy broadcast, you can call 800-732-2334. Today's show is number 1052. That number again, 800-732-2334. You can also find it on our website, peoplespharmacy.com.
And when you visit our site, you can check out information on prescription drugs, herbal medicines, and home remedies. Share your thoughts on today's show. Have you experienced antibiotic resistance? We'd love to hear your story.
At our website, you could sign up for our free online newsletter. It comes twice a week to your inbox, or the free podcast of the show. You could listen at your own convenience or share it with a friend. It's all there at peoplespharmacy.com.
In Durham, North Carolina, I'm Joe Graydon.
And I'm Terry Graydon. Thank you for listening. Please join us again next week.
We hope you enjoyed this podcast. If so, please consider taking a minute to write a review on iTunes. And thanks for listening to The People's Pharmacy.
If you're running a retail business, don't let disorganized order fulfillment cause chaos. Use ShipStation instead. From running a business out of your garage to multiple warehouses, ShipStation is ideal for every phase of your growth. Save time with one login for all your stores and by automating tasks. Plus, you'll get the best shipping rates from global carriers. Calm the chaos with the shipping software that delivers. Start a free trial at ShipStation.com/audio. That's ShipStation.com/audio.